we could not only measure how fast we are aging, but actually test whether lifestyle changes or supplements could slow the process down? Aging clocks are starting to give us those insights. And one of the pioneers at the intersection of science and interventions is our guest today. Hey everyone, welcome back to Decoding Longevity. I am Macsue Jacques as a host today. And in our last episode, we have explored how precision medicine, geromedicine is moving aging research from lab into the clinic. Today, we're going to take a step further, diving into a landmark paper in NPJ Aging titled LeanAge 2, providing actionable insights and benchmarking with epigenetic clocks. And to help us break this paper down, we're joined by none other than Professor Brian Kennedy, distinguished professor at the National University of Singapore, director of Healthy Longevity Translational Research Program, and a global leader in the biology of aging. Brian, welcome to the podcast.
Great to be here. Thanks for inviting me.
Thank you. Okay, so your paper introduces the LeanAge, which is LeanAge 2, which is the update from LeanAge 1. For the sake of our speakers, and just to give a little bit of how this clock is actually different from, let's say, the Horvath clock or the PhenoAge, what is actually that you had in mind when you created this clock?
Yeah, I'm happy to do that. First of all, I'd like to just point out that Jan Gruber and Feng Sheng in US are two great scientists that had a big role in building this clock. So my job is mostly to take the credit. They do a lot of the work. So I need to point that out. What we were trying to do, and I should say there are many, many clocks out there. Most of them are DNA methylation-based clocks. And they're very interesting. They're first generation clocks that try to predict your chronologic age, and second generation clocks that try to predict an outcome like mortality, and then other kinds of clocks as well. I think the challenge we had with it is that when we're trying to go into the clinic and actually change people's lives, it's very hard to do that with methylation. There are several reasons for that. One is that clinicians are not taught to really understand epigenetics and DNA methylation. Some of them really have a great grasp of that, but most people in hospitals are not thinking about that kind of modification. In addition, it's not a standardly used measure, so it costs more to measure your DNA methylation, and there's more noise in the measurement because the standards of how you measure it are not as determined as they are for something like clinical parameters. So what we wanted to do was to take modifiable clinical features, things like LDL, HbA1c, your blood pressure, use those as features, and then try to see if we could build a biologic aging clock that predicts mortality from that. The reason is that those measurements are standardized. They're measured more or less the same way anywhere with very little noise in the measurement. In addition, people understand those measurements, and maybe most importantly, they're modifiable. Almost everything we measure is modifiable by a lifestyle change or a drug currently today. So when you get a measure of your mortality using our LeanAge 2 clock, what you find is that you can change it. You can reduce your mortality risk by taking well-known medications that already exist. So we built this clock from a retrospective data set in Haynes, which in the US, which measured what happens to people 200 months from the point of measurement. So we can predict mortality from this clock. And now we're using that in the clinic to try to see if we can modify a person's mortality risk.
Yeah, that's very interesting because it becomes much more clinical relevant and you can actually action on something. So I wonder, did you find as well that you could predict somewhat the development of disease, not just mortality? Could you, for example, say that someone is likely to develop, I don't know, high blood pressure or, a cardiovascular condition by just looking at this clock? And could you then use this as a prevention and start treatment even before the symptoms appear or the markers are actually out? Is that a possibility?
Yeah, that's exactly the idea we're thinking about. So there are clocks where you can try to measure specific disease, or you can try to measure how fast a particular organ is aging. We chose not to do that because I fundamentally believe that there's a aging process in the body that's more systemic. There's a homeostatic mechanism in your body that tries to keep you healthy as long as possible. And we wanted to measure that sort of as a top line measure. But what we did is use principal components to generate that number. So then we can go into each principal component, look at the features of that component, and get an understanding of what's driving that particular risk in that feature. So some components are composed of features that are associated with metabolic disease and others, with lifestyle events like smoking. So when we break those components down, we can ask what individual factors are driving this person's accelerated aging risk, and then go inside of that parameter, look at the features and try to modify those features. And even better, we can predict what's going to happen because these, like I said, if we stick to clinically approved drugs on label, then, we know how fast those drugs are likely to act on average in a population. So we can look at, say, 1,000 people and say, all right, in this population, we're confident we can bring down the mortality risk by X amount, which equates to 5 years younger in your biologic age or something like that. It depends on the population, of course. And that's only using standard on label medication. Of course, we would love to go next to testing geroprotectors and other kinds of longevity interventions, which may have an even broader effect.
Yeah, I guess that brings to a key question. Once we know what is actually wrong, then we can act on it. But then there's also a lot of research going about, as you said, protectors or even supplements that might actually slow down aging. And I know that you presented something at ARDD, and it was really, really interesting, where people are commonly hearing about all those supplements and they just go for it and without really knowing what is actually wrong. And sometimes combining some supplements may actually, inhibit the actual potential of that supplement, for example. So I guess I have two questions in that regard. Are we ready to use this clock in the clinic? And, what would be the next steps to be implemented in clinic? And the second is, do you think, how can we then convert the knowledge that we're getting from it to then start using those geroprotectors? Do we actually have enough data to go and give advice to people, hey, you should take such supplement or such medication?
Yeah, well, let me take the second part of that question first. We are using it already in the clinic now. I'm not here to say that it's fully validated. We're using it in a research strategy to try to get that validation now. And the thing I like about it is that I'm not an MD, I have to say that ahead of time, but I'm meeting people that are using the clock. And so we're getting all the numbers and all of their health behavior and then talking to them individually. And I think you learn a lot from those sort of N=1 case studies as well. I think that we get to see how well we think things are working by understanding what's happening to people. And so I feel like we're definitely on the right track. Now, I'm not going to say that we perfected this clock because people might have rare conditions that alter the clinical numbers in odd ways. We're still trying to look for those. And in addition, we're probably not capturing every single facet of aging yet. So we're continually trying to improve the clock, adapt it to different ethnicities, that sort of thing. But I feel like we're definitely on the right track and we can have an impact now. If I could come back to the supplement question, that's the next thing we want to do is test supplements and drugs in the context of this. Now we can already kind of test drugs and supplements to some effect because we can go look at UK Biobank data or other types of data, and we know what medications people are taking. So that gives us a retrospective sort of analysis of which drugs are affecting aging. And we're starting to see some of the leading candidates have an effect on aging using those studies. What we need to do is test this prospectively, ideally in Singapore, but also in other locations, to see whether we can move that needle with geroprotectors. And I think things are going to be extremely personalized. Nothing is going to work in everybody, even exercise. Some form of exercise might benefit everybody, but telling everybody to run marathons may help some people and hurt other people. So what we need to do, even in that case, is understand the right level of exercise. And I think that's going to be true with supplements. Which supplements benefit which people? We're already starting to see that in some methylation data we looked at. And we really need to connect these dots. We're getting great diagnostics of aging now. We've got many candidate interventions to slow aging. And we need to understand which ones are affecting which clocks, and maybe more importantly, what are the functional outcomes of that.
Yeah, I guess that's super interesting because years ago I actually run individualized analysis of exercise training, and what I have seen is that by applying the same exercise to a variety of people, some would respond really well in some areas, but not
Yeah.
all of them. And even exercise is actually really personalized, and I think we have to stop thinking just reducing everything into the mean and considering everybody as a general population, and really focus on gathering this longitudinal data where we're actually now able to do this personalized or one type of trial. So that's really exciting. But I guess
I know one thing though, that with all the travel I've been doing, all the conferences in the last week, I've been applying no exercise to myself. So that's a bad habit. Today I'm going to fix that. So
yeah, that's good. That's good. No, that's all right. I guess it's all, as long as you catch up later on as well. I think people are too hard on themselves as well, or like, oh, I didn't exercise for 2 days, or I had too many drinks the other days. I think it's as long as it's not a constant thing, I think we can always catch up. That's the beauty of being human, right?
we need to
Um,
always wake up and thank resilience.
Yes. Yes, minor stresses are good. But I guess going back to the clock itself, and seeing that it's looking into individual diseases and potential group of biomarkers that are changing, that are likely to develop into one disease. Would you agree that then we're going back into sort of like the normal way of seeing things, where we're targeting individual diseases rather than targeting aging itself?
Well, I think we're at the interface with this clock now. I would never argue it's the most mechanistic clock on aging, looking at the core changes that drive the beginnings of the aging process. I think that's really important, and it's something we need to use potentially proteomic and metabolomic clocks for. So no clock serves every purpose, but those clocks are not easily actionable right now in the clinic, and we're looking at the changes in aging that are beginning to drive disease progression. We're not measuring disease really, we're measuring markers or risk factors of disease. So what we're trying to do is get earlier in interventions. And as we do that, I think we're making the transition from disease to aging. And so we're probably more on the aging side than I think most biomarkers are in terms of action, in terms of ones that are clinically actionable. Hopefully this is a transitional thing. What I'd love to do is be able to measure people at 25 or 30 and say, hey, you've got these proteomic changes and
Mm,
metabolomic changes, and we know where you're going to be in 30 or 40 years, and we know what you can do now to not get there. That would be great. I don't think any
mm,
clock can do that right now.
yeah.
And so we're going to continue trying to optimize the clock we're working on, and work that process back further and further, so we, have indications what people should do throughout their adult life.
I guess like this really leads to the next question, where you said, not one clock suits everything. And another work of yours that has been recently published is the Dolly clock, which is the aging clock using lipidomic profiles, which is quite different from DNA methylation. So I guess what made you think and turn into lipids as a basis of building this clock, and why is it so linked to, Alzheimer's, for example?
Yeah, that's a great work by master student Jacques in the lab. He's still working in our group with Jan Gruber now. So we've got a great team of sort of dry lab researchers. I think that my feeling is that, well, there's some people that believe epigenetic changes of aging are the drivers of aging. And if that's the case, you're going to the source by looking at epigenetic changes. But on the other hand, you can take any complex data set and try to measure the person's age. And we're excited about things like proteomics and lipidomics and metabolomics, because these are the molecules that are doing things in the cell. Changes in DNA methylation are affecting gene expression and maybe other nuclear events. Um, but it's hard to figure out how they're affecting gene expression. And even if you know that, you're going from DNA to RNA, and then you still have to make proteins, which are highly regulated. So I don't think it's the best direct measure of the mechanistic changes of aging by using methylation. If you measure like 20 proteins that change that predict your age, then most people, we can look and figure out what most of those proteins are doing. So we have some idea of which hallmarks of aging they're impacting, what effect on the body they're having. So we've been turning to proteomic clocks and also metabolomic and lipidomic clocks to try to measure aging. Lipids too are very actionable molecules in the cell that are doing different things. Now, challenge with lipidomics, especially, is that it's hard to get consistency from one lab to the next in how you measure lipids. And so I don't think this clock is ready for clinical use right now because lipidomics is not that consistent in the experimental process. With like I said, we different clocks have different purposes. We built this clock for a different purpose to try to understand how lipids are impacting aging. And the other challenge is getting lipid samples. You can do that from blood, and we have another story coming out on the relationship between aging and pancreatic cancer using lipidomics. But this one, we were able to get access to a data set where they measured lipidomics in postmortem brain samples. And so we wanted to look in brain tissue and try to understand aging. So it's really hard to get samples of that. So we didn't have a huge number of samples, but we had enough to build a clock to measure. So we're trying to predict aging, a person's age from their brain samples. So it's kind of a combination of what's happening in the brain and maybe some systemic signaling with the rest of the body. And what was striking is the algorithm picked a certain class of lipids that could predict aging, these dolichols, which are involved in leak-like oscillation. We didn't try to focus on those molecules to start with. We took all the lipids we had, but the computer said we get the best prediction just by looking at this class of lipids. And so that points to their relevance in brain aging. And I think it's also reassuring because there are papers 20 or 30 years ago that had already noticed that at a more less refined level, they had noticed that there were changes in these dolichos with age. So again, we're using sort of these black box AI approaches, but it's bringing us back to mechanisms of aging that we know about. So it's very reassuring. I think there's a ton to be learned from lipids. There are literally millions of lipids. You have probably 100,000 of them in your body, and only a small percentage of them do we really understand what's going on. Some of those, though, are really important ones, like estrogen is a lipid, and many other things in the body that are having huge impacts on our aging process. Some of the things we put in our body, too, are lipids, but maybe I won't go into specific classes of those molecules.
No, that is very interesting, and that's for sure, something that could reveal a lot of differences, especially, since you mentioned the estrogen, differences between males and females, and because males and females accumulate lipids throughout life and also metabolize and, use those hormones and are lipid dependent, in different ways. So I guess it will shed a lot of clarity into that sex specific aging processes and the changes
Yeah.
that are happening.
It's really an area of biology that we don't know enough about. If we can really learn more about what lipids are doing, I think it's essential to understanding how we function. And like I said, like you said too, some of the lipids, like hormones, we study a lot, but most of them just are, we have only a vague notion what they're doing.
And I guess like you also mentioned, the protein clocks coming into surge now. And it's funny because the last, say, prediction to mortality was the LeanAge in 2016. And since
Yeah,
I feels like there was sort of like a stagnation where all the attention was, directed to DNA methylation and very little was done in the proteome world. And it feels like we got stuck into that first generation clock where we really just predicting, chronological age and other than the LeanAge, maybe I am unaware of any other literature predicting, biological age. And I think
yeah,
it's time really that we do that jump. So I guess there is a lot of discussion on like how trustworthy the clocks are in the
oh yeah,
sense that they are not necessarily capturing in a live manner changes from interventions. So, for example, someone does calorie restriction or does exercise, it doesn't necessarily move the needle of the clocks. Some people are taking that, oh, it's not actually changing your biological age. But what if the clocks have reached certain accuracy? And I'm here just to sort of like tease out with this question where what if it's the other way around? The clocks are really measuring accurately, but really it takes so long for those interventions to actually do a significant effect where these changes are now reaching a point of detection. What do you like? Do you have any insights on this, and what is your thoughts?
That's a challenge, right? We're still connecting the dots here. And so we don't really know how long interventions take to work in humans. We have an idea in animal models, and often we can see changes in trajectories in mice in 3 months when we put them on an intervention. This paper that we're working on now, where we're using a relatively simple methylation clock and we're measuring changes in supplements, we can see changes in an average of 4 or 5 months, and that's methylation. So I would guess somewhere between 3 to 6 months, but different interventions may take longer periods of time to work, and we just don't know that. So you're right. When you give people interventions and you don't see a clock move, you don't know if you haven't waited long enough, whether the intervention is not working, or whether the clock is not capturing the changes that the intervention is making. And I think these clocks are still a work in progress. And if any particular clock, even ours to some extent, it may not capture every aspect of aging. So if you have an intervention that's in this area and your clock is capturing things over here, you may not see the effect of that as much. And I do think when we're trying to show efficacy in people, we should be doing our best to use these clocks to predict outcomes. So, like LeanAge you mentioned, or LeanAge too, they're trying to predict some outcome like mortality, so or some set of clinical parameters associated with aging. So, I think that those are the best ones because we know when we see a change that it's linked to an outcome. I think it's harder to figure out when you see a change in a clock that's just predicting age what that means. Now, the lipid clock we just talked about, we predicted age. We didn't have mortality data. These people were mostly people that suffer from accidents. Sometimes you're limited by the kind of clock you can build, but I think that if we can, the second generation clocks that predict outcomes are much more comforting to use in a clinical condition because you can convince yourself you're really having an impact if you change the needle.
So you mentioned a combination of supplements, and I know that you're doing some work in looking into some supplements, which is great because I feel like sometimes people just take everything and they think the more that you do, the better it will be. And it's you seeing that that's actually not the case. So would you mind talking a little bit about your findings and the supplements that you're working with, and sort of like what are the ones that you think hold the most potential, if you're allowed to?
Um, First of all, we've done a lot of animal studies combining different supplements together. And the take home message is that sometimes they're additive, but most of the time they're not. So, and we don't know when it's going to happen. So, I really caution people against trying to take, 20 or 30 or 50 or 10 supplements because we don't know how they're interacting with each other. And a lot of interventions have sort of convergent properties in cells. A large number of interventions impact the mTOR pathway, for instance. And so this pathway is activated by nutrients or stress conditions or infection, and it needs to be activated. It has a role in that process, but it's also turned off during a resting state in the cell. And when you're young, you have a good dynamic range. You turn the pathway off when you're supposed to, and you turn it on when you need to. As you get older, it looks like that off state doesn't work anymore. And so it's coming back on. And so when you take rapamycin at the right dose, you can sort of restore that dynamic range. At least that's how we think about it. But if you're taking rapamycin and six other things that impact the mTOR pathway, you might be affecting its ability to be activated as well. And then that's going to cause a lot of other problems. So these interventions don't all work in silos. They interact with each other as well. And we need to understand which ones work together. So I tell people, look, I think there's some of these things are benefiting people, but you have to understand which ones benefit you. So first of all, don't take 10 things at a time. Second of all, as if you're taking something, measure its effects. I know it's expensive in some cases, but it's possible to
Mm.
measure these clocks. You can go to the doctor and measure your clinical parameters. If you have high glucose, you can see what it's doing to your glucose, for instance, and see what works for you. I find that I go on things, go off things, combine a couple things together. And I found combinations that seem to work really well for me. But whether that's the ideal thing for you or not, I think you have to find out yourself. So I tell people don't just take stuff, measure things as much as
Mm.
possible. And I know this is hard, right? If you're not a
Mm.
scientist, it can be complicated. But at this point, we don't it's not easy. I can't even tell you immediately what combination is going to work in you. So if you're going to be effective, you need to be proactive and inform yourself and, do those kinds of experiments safely to see
And
what's helping you function better.
yeah, absolutely. And I guess like another issue that there is as well, even for who is a scientist, yes, as you mentioned, it can become quite expensive. Like I recently went to the doctors and I asked them those parameters measured and they looked at me and like, oh, this is not covered by Medicare.
Yeah,
Oh,
yeah,
we don't
exactly.
do this. We have to find another place. And so there is like so many lines of like resistance for you to actually just get an answer and just be able to measure
Yeah.
yourself. And I think that's a more deep rooted issue in the system that it needs to be changed. And this way, doctors now potentially could be prescribing at least an annual checkup. Hey, are you done this year? This is working or not? And just be able to guide people. Hey, this is actually not being good for you or not. Or the other way around, it might be going great. So yeah, I think there's a lot that still needs to be changed.
Well, I think you touched on a key issues there. I mean, first of all, before I answer this question in a way that makes everybody think that I'm just out to help billionaires, let me first say that we need to democratize this process. We need
Mhm.
to find strategies that are accessible to everyone, hopefully that are reimbursed. Believe me, if we can slow aging and reduce mortality risk by 2 or 3 years even, and I think we can do way better than that, but if we can
Mhm.
do it by 2 or 3 years, it's worth spending the money on these tests. The amount of money that you would save as a company paying for your insurance or as a government that deals with public insurance is in the billions of dollars, at least for the governments. And so it's worth it to do this. But right now, you're right, it's very hard to get a lot of these tests. Now, doctors can generally, it's usually sending blood or urine or something that's not that hard for doctors to do, but they're not used to people coming in and saying, I want to pay for this part myself, even if I can't get reimbursed.
Mm.
But you can find clinics that will do that, and even standard doctors will often do that. You may have to
Mm.
convince them why you want the test. But right now that's where we are. If you really want to get this information, you probably have to pay for some of it, and if you have the resources, I think it's definitely worth it because I think we're learning how to do this process right now. We're learning how to slow the aging process. And there's information out there that's sufficient to help most people already. Now, how long it takes to get that into standard medical care, I'm doing my best, and I know a lot of other people are, but it's an uphill battle, so it may take a while.
I guess like yeah, just pushing this into the clinic is another issue, right? Because the medical system is just so used to treating the disease that when you talk about prevention, not it's sort of like out there, and if it's still out there, even though it's so in your face all the time, like prevention, take care of your health and all the things. When you actually go to the doctors, they usually ask, okay, what is wrong? Not like, okay, how can I
Yeah.
potentially help you not to get to that point? So.
And then also the doctors are really trying to help, but you have to understand their perspective too. They went through medical school, which in years in training, they learned probably nothing about aging. It's unbelievable that aging is not in the curriculum. It's like one of the few things that's happening to every single person
Yeah.
that walks into the doctor's office. And we know it's a risk factor for a
Mm.
whole range of different diseases, and yet it's still largely ignored in medical schools. And we need to get out of the 19th century in how we teach medicine.
No, absolutely. Brian, this has been like so much fun. Thank you so much for joining the podcast today and helping us understand a little bit more about the science around the clocks and the potential new interventions that are coming. To the listeners who are interested in Brian's work, we're gonna put the links for the LeanAge 2 paper and the resources for Brian's work in the notes. So if you like the episode, please make sure to like, share, and tune in for the next time. Thank you so much again, Brian.
My pleasure. We'll discuss the next advances in the bar at the next conference.
That sounds much more fun.